Good morning, ladies and gentlemen, or good afternoon, or good evening, or good night, whenever you may be listening to this. So this is going to be the recorded lecture for the virtual day on the 16th of January. So go ahead and listen to this, take notes as you see fit, and then I'll be posting a CK12 actually two CK12s on the Google Classroom as part of the attendance assignment. All right, so last time we left off kind of talking about phylogenetics and how we can look at either genetics, we can look at shared characteristics, we can look at the proteins and compare relationships between different species. So that was kind of on the really, really micro or molecular level. Now we're going to take a step back and look at kind of the larger level, specifically when we talk about biogeography. Now, biogeography, if we break that word down, bio means life, geo means earth, graphy means drawing. So essentially this is just the integration of mapping where things are at within the world, including that into kind of the evolutionary history of organisms and biology as a whole. All right, so biogeography is a study of the distribution of plants and animals across the world and helps explain how geographic changes influence the evolution of species. So some of you may remember from earlier science classes about Pangea, where all the continents were combined into one supercontinent, and South America was kind of paired up with Africa. And during this time, one thing that we start to notice is when we look at the evolutionary history of certain organisms, you'll see similar fossils on the eastern side of South America as you do on the western side of Africa, showing that at one time when these were hypothetically or theoretically connected, that you saw similar species living in the same area, so you would see the fossils between them as well because they used to be connected. All right, when we talk about biogeography, we'll kind of use camels as an example. So these originated in North America about 1 million years ago, even though they are not present here now. And over time, they migrated to East Asia via a land bridge during the last ice age. Some reached Africa, others migrated to South America via the Isthmus of Panama. And essentially what occurred here was independent evolution, where in different environments, camels evolved adaptations suited for those specific areas. Now, a lot of you right now think camels, deserts, and so a lot of the camels that we think of now have certain adaptations for those types of environments. But geographically, it looks something like this, where camels originated in North America. Some migrated south to South America. The environment there kind of served as selective pressure to create llamas, which are related to camels. And then from there, the ones that migrated north across this land bridge between Alaska and kind of Siberia or eastern Russia eventually made their way down to Asia. The Asian ones became the Bactrian camel or the two-humped camel. And again, some of them migrated all the way to Africa. These are the ones that are going to live in the desert, pretty harsh environments. And this is the dromedary camel. So you can see how they all look a little similar, but each one of them have their own adaptations for the specific environment that they are in. You can actually track some of the fossils as they start in North America and go to other parts of the world. So, island biogeography. This is what Darwin observed during his voyages on the Beagle. And it provided strong evidence for evolution. So if we look at Darwin's finches in the Galapagos Islands, they all likely descended from a single seed-eating bird species. But because each island had its own little environment, that single seed-eating bird species evolved or adaptively radiated into many species with different food specializations. And you'll see kind of a visual of that on this next slide. Right, now the differences between these species is really evident in their beaks. They had different sizes, shapes, adapted to specific food types. And we're going to see that figure on the next slide here. So the central finch is going to be the common ancestor. And then depending on what type of environment these finches were in, they adapted and kind of evolved to have different beak shapes suitable for whatever type of food they were eating. So the ones eating insects had a little bit narrower beaks. Grubs had even narrower beaks. If it used tools, is going to have a harder, tougher beak. And then buds and fruit kind of had this shorter, rounded beak as well. But again, all these differences are created by the selectional pressures due to the different environments on each of the islands that these finches were on. And we call this adaptive radiation, because a single common ancestor kind of adaptively radiates into multiple species of finches. So when we talk about adaptive radiation and changes over time, which that's what evolution is, we're going to look at something called the Grant study. So you had these two researchers, Petey and Rosemary Grant, and then over a 30-year period, they studied finches in the Galapagos Island. This is the same place where Darwin was looking at his finches. They observed kind of these changes over time and noticed changes in the finch populations during a drought. Remember, this drought would serve as a selectional pressure. And then birds with larger beaks survive better, leading to an increase in average beak size in population. And this demonstrated evolution by natural selection in action. So again, this is going to be similar to the giraffe example we talked about in class the other day, where the individuals that are better suited for the environment at the time would survive better, produce more offspring, therefore those genes would be passed on to the next generation. So this is actually their data from their 30-year study. And they actually looked at just 2 years during which a drought occurred, and they noticed that the average beak size increased over this 2-year period. And if you think about it, 2 years within the grand scheme of biology and life is not very long. And so we saw a change from around 9.3 to 10.1 in beak size within just 2 years of these birds during this drought. This video real quick.
Next up, biogeography.
Well, biogeography is the study of how animals, plants, and other organisms have come to occupy the places on the globe that they do.
Imagine a new island being born in the middle of the ocean. At first, it's a hunk of rock, lifeless, but gradually, seeds and insects and small animals originating from nearby land masses get blown over on the winds. They come crashing ashore, pushed by the waves. Not all the organisms make it, but those that do, with time, adapt to island living, perhaps even evolving into new species. Just take birds. There are the finches of the Galapagos Islands, the birds of paradise of Papua New Guinea, the honey creepers of Hawaii, and so on. In each case, a single island species diversified into an array of species found nowhere else on Earth. Biogeography's also got something to say about why some closely related species flourish on different continents. It seems odd until you recall that Earth didn't always look this way. A few 100 million years ago, all land was part of Pangaea, a hulking supercontinent. There were no vast oceans to interfere with the movements of organisms. But then, starting about 170 million years ago, the continents drifted like vast rafts across the sea. And the species living along the edges, they were split in two. Fast forward to the present day, and you get a plant in South America whose close relatives grow in the tropical Pacific.
We can use phylogenetic trees as sort of maps to help us reconstruct the movements of organisms across the planet.
All right, so a few practice questions. So which statements are correct for Peter and Rosemary Grant's study on Darwin's finches? A, birds with bigger beaks survived the drought and reproduced. B, birds with smaller beak size survived the drought and reproduced. C, after 2 years of drought, the average beak size increased. Or D, the Grant study could prove the evolution of finches by natural selection. Think about your answer now. So the correct answers are A, C, and D. The birds with bigger beaks survived the drought and reproduced because they were able to feed on seeds of all shapes and sizes and were able to survive and reproduce more compared to birds with smaller beak sizes. After 2 years, C, after 2 years of drought, the average beak size increased. If you notice from looking at the figure 4 earlier, you saw the average beak size increase from about 9.3 to 10.1. And then because they were able to see this, D is also true, Since the beak size increased, birds with bigger beaks survived, reproduced, and were naturally selected, showing that change over time due to natural selection. The above graph represents Peter and Mary Grant's study of Darwin finches on the Galapagos Islands after drought. What inferences can be drawn based on the above chart? A, the population of finches decreased. B, the average beak size in 1976 was 10.1. The average beak size of finches increased in 2 years. Think about your answers now. All right, the correct answers are A and C. So if you look at this graph to the left, so this is called a histogram. It basically looks at the prevalence of individuals with relative beak sizes, 8, 9, 10, and so forth. And you can see if we look at the 1976, there's a lot more area covered by these lines compared to 1978. This indicates that the population size went down and some organisms didn't survive. Now, as you notice here, most of the organisms' population that didn't survive were left of this line. So if you look at where the averages were and where the average is now, you can see that the individuals to the left of that average in 1976, there's not as many of them, right? And then the average beak size increased in 2 years. You can just see where they have the averages between these. And obviously this is driven because a lot of these finches with smaller beak sizes didn't survive during the drought. They didn't have as many seeds to eat, etc., so they died out. What is an example of adaptive radiation? A, finches found in Galapagos Islands with different beak sizes and types of food they eat evolved from a common seed-eating ancestor. B, plants of the same species growing in different parts of the country or region. Or C, vestigial organs like the appendix and wisdom teeth in humans. So the correct answer is A, finches found on the Galapagos Islands with different beak sizes and types of food they eat evolved from a common seed-eating ancestor. Okay, so B is just kind of an example of biogeography where Pangea spread apart, and that's why you see same species growing in different parts. And vestigial organs are kind of organs that are left over that aren't used anymore. But that is not an example of adaptive radiation. So in summary, biogeography shows how geography affects species distribution and evolutionary paths, and provides examples like camels and Galapagos finches to illustrate these processes. Adaptive radiation was when one species evolves into multiple species to fill different environmental niches, and observed in island ecosystems like the Galapagos. All right, on your own time, go ahead and try to mentally go through these review questions, see if you can answer. Review the slideshow or the CK12 section if you do not understand something, or, as always, reach out to me by email. All right, population genetics. This is just studying the genetics of a population. So genes with a G are hereditary, while genes with a J are not. So when we talk about gene pool, we're talking about genes. Remember, genes code for proteins during that central dogma of molecular biology. And then when we talk about evolution, it's just change over time. And specifically, it's the changes in gene frequencies within a population. Now, if we're in school tomorrow, we'll talk about something called the Hardy-Weinberg equation, which actually looks at and calculates how gene frequency changes over time. And that is really the measurement of if a population is evolving. All right, so Darwin recognized the need for heritable variations for evolution. Now, these variations are a byproduct of mutation in these genes, causing variation and changes within the offspring so that all your offspring are different, along with the other things that cause your offspring to be different and results in genetic variation, random assortment, crossing over, random fertilization, etc. When they kind of looked at Mendel's laws of heredity, it kind of helped provide the genetic basis for understanding what evolution is. And then these heritable traits were caused by different alleles. So if you look over there to the right, you've got different alleles for different, we'll say, hair colors of gophers. And then apparently the soil was very, very dark, so that darker gophers were able to blend in a little bit better. Therefore, they were not as easily identified by predators, and so the darker gophers survived. All right, now when we talk about evolution as a time scale, we're going to break it down into two. There's microevolution, which is the short-term evolution within a population or species. And this is going to be exemplified by adaptive radiation. So kind of Darwin speciation, where you go from that common seeding ancestor of finches, and then you see very, very closely related other species that happen within a relatively short time. Okay, and you've got macroevolution, which is evolution occurring over geological time above the species level. So this is going to be something like where we go from the first eukaryotic cell to all of eukaryota in general, plants and animals. Microevolution has been observed by the Grants and Darwin's finches over time, over a short period. Remember, this is where the beaks got longer or larger due to the natural selection from a drought. And then macroevolution is reflected in the fossil record, resulting from extended microevolution. So they kind of go hand in hand. If you look on the visual to the right, you kind of see the evidence in the fossil record from kind of ancestral horses to modern day horses, and how the molars have changed as well as the bones and then the size as well. Now, when we talk about evolution, the unit of evolution is the population. Individuals do not evolve, populations do. This is a key statement that you should be aware of. And then population genetics is just study of evolution within populations, integrating evolutionary theory with Mendelian genetics. So you can look over here to the right, and this is just kind of an example of population genetics and microevolution in practice. And so you have a parent population, let's say a portion of it gets separated either by an earthquake, a river comes through, or they just migrate to another area. But they're in a new environment, and so the old selectional pressures of their old environment aren't present in the new environment. And so the population will evolve so that after a few generations, the frequency of genes has changed just over that time period. The gene pool is a combination of a couple things. So the genotype and population, the individual's genetic makeup is their genotype. A population has many genotypes. Remember, that's the combination of those two alleles in most traits, or several traits, I should say. And the gene pool just comprises all the genes of all the members of the population, including all the alleles for each gene. Now, it's important when we talk about this, we're looking at the genotype, the actual genetic makeup of these organisms, not the phenotype, not the physical representation of those genes. So over here we have big R and little r. If you count all the big R's, you'll find that there's 20 of them. If you count all the little r's, you'll see that there is 10. So when you look at the allelic frequency, the frequency of R would be 20 out of 30 because there's a total 30 genes. So you get 0.66. The frequency of the little r, or the allelic frequency of little r, there's 10 of them out of 30, so its allelic frequency would be 0.33. So allelic frequencies are the frequency of occurrence of alleles in a gene pool relative to others. So this will always be represented as either a fraction or a decimal point, pretty much exclusively below 1. It will never be above 1. For example, let's say we have 200 gene copies. So that's based on 100 individuals, and remember, each individual has 2 copies of each gene, one from the mother, one from the father. So if we count, we'll use the letter A. So if you count the big A's and the little a's from the different genotypes, big A, that one will have 2 big A's. Big A, little a has 1 big A, 1 little a, and little a will have 2 little a's. So if we want to calculate the frequencies of P for A and Q for little a, when we talk about P's and Q's, that's the Hardy-Weinberg equilibrium, which we'll get into in the next section. We can do the calculation like this. So for our genotype, we've got them listed here at the left. Big A, we have 50 of those individuals. Now we're going to multiply that 50 by 2 because those with this genotype have 2 copies of that big A gene. So that means we have 100 copies of this big A gene here. Okay. All right. Now big A, little a, there's 40 individuals. So this one has 1 copy of the big A, 1 copy of the little a. that means we have 40 big A genes for our heterozygous individuals. Now for our little a, it only has little a's, so we have 0 big A genes. If you kind of add them all up, what we see here is we have 140 big A genes. And since we're only dealing with 2 alleles, big A or little a, the remaining genes will be little a for a total of 200 genes for 100 individuals. So if we have to look at the frequency, so the question would be, what is the frequency of the big A genes? And we just do 140 divided by 200, because that will be the total, and that would be equal 0.7. Conversely, what would be the frequency of little a's gene? We do 60 divided by 200, which would be 0.3. Now, if you notice, all of our frequencies, when added together, will always add up to 1. So this is just kind of a line-by-line version showing the calculations and the numbers that have to go into calculating gene frequency, or allelic frequency, excuse me. And remember, P plus Q will always equal 1. Now, the reason we don't say big A plus little a always equals 1 is because sometimes you're going to have that codominance where it could be big A, big B, so on and so forth. So we just use the letters P and Q to signify one allele or the other. All right, if you want to see a visual of this little demonstration, you can go to that CK12 section in your digital textbook, and you can actually see the visual show over time. But essentially, we've got these population of birds. We've got big B and little b. The big B dominant gene codes for brown color. The little b codes for a yellow color. So we see our allelic frequency here toward the bottom right of our initial population. So for big B, it's 0.4. And for little b, it is 0.6. Now, one thing I want you to notice is that even though the little b allele has a higher frequency, if we look at the phenotypes of our population, there's 1, 2, 3, 4, 5 finches or birds that show the kind of the yellow coloration, which is the little b. And then there's 1, 2, 3, 4, 5 that show the big B genotype, or big B phenotype, excuse me. So just because we see a higher frequency of little b within the population doesn't mean that that phenotype will be at a higher percentage. And this really has to do because the big B is dominant. And so your heterozygous individuals, for example, this one and this one, they will show the dominant trait and kind of throwing off your math here. Now let's say if there's a predatory bird population or species that comes in and it starts hunting down these species, and it targets either the brown ones or the yellow ones. Let's take a look. So target frequencies in sparrow population due to external factors like predation and parasitism. This will impact the allele frequencies over generation. So if we have a predatory bird come in, and this predatory bird can see these brown birds a lot easier, that'll be a natural selection against brown and a natural selection for yellow. And you will see that the little b frequency increases over time, right? We are changing the genetic makeup. We're changing the gene pool of this population. When we talk about evolution, that's what it acts on, is a population over time. So we see a decrease in the big B allele and an increase in the little b allele over time due to the selectional pressure from the predatory bird. Now, let's say we have a parasite that for whatever reason more likely kills or attacks these yellow birds. We see the opposite, where we see a decrease in the alleles for yellow, the little b alleles, versus the other, the brown. So we'll see an increase in the brown birds and a decrease in the yellow birds. Now, one thing you might notice is that these changes aren't as drastic when compared to selection against the dominant allele. And that's because these individuals that are heterozygous, they get the same benefit theoretically as the individuals that are homozygous dominant. And so you can still carry on these little b alleles to the next generation because they aren't selected against when they are present in the heterozygote. All right, some practice questions. An insecticide that was known to kill a specific type of beetle was suddenly found to be ineffective in a small group of beetles in a population. Over the few generations, this was due to resistance developed by this group as a result of genetic mutation. This is an example of macroevolution. Think about your answer now. All right, the answer is false. This is microevolution, heritable change that occurs over a relatively short period of time, unlike macroevolution, which becomes noticeable after many, many generations. Think hundreds of thousands of years. Humans and chimpanzees have evolved from a common ancestor about 6 to 8 million years ago. What type of evolution could result from this? This one's going to be macroevolution that occurs over geologic time above the level of species. This is basically results from microevolution over many, many, many generations. A population of 300 sparrows has 150 individuals with red beaks, big R, 60 individuals with brown beaks, little r, and 90 individuals with reddish brown beaks, big R, little r. What is the allele frequency of little r in the population? Think about your answer now. Now remember, a hint, we have 300 sparrows. Each sparrow will have 2 copies of each gene. That means when we're doing our math for allelic frequency, it will be out of 600, not out of 300. So if you got your answer as 0.35, that is correct. So in summary, microevolution occurs over a short period of time in a population or species. Macroevolution occurs over a geologic time above the level of the species. Population is the unit of evolution. The gene pool would consist of all the genes of all the members of the population. And then for a given gene, the population is characterized by the frequency of different alleles in that gene pool. Go through these review questions on your own, see if you can answer them. If you can't answer them, please review either the slides, go through the CK12 in your textbook, or shoot me an email as you see fit.